My name is Pan Panjaka. I am a scientist working for the Anti-Cancer Fund based in London but working for a Belgium organisation. Drug repurposing is taking an existing drug and using it for a new illness or a new indication. Specifically, I'm working in a project called the Repurposing Drugs in Oncology project, which is looking at taking non-cancer drugs, common drugs which are used for many different diseases, and using them to treat cancer as well. The advantages of drug repurposing are that we have a huge history of using these drugs. So we know how these drugs work in patients treating the original disease that the drugs were designed for. We know the common side effects, we know the rare side effects, we understand how these drugs work and which mechanisms are important to how they work. So we can take all of those advantages and apply them to cancer. We also have the advantage that many of these drugs are generic, so they're very cheap, and they are generally not very toxic, unlike many cancer drugs. These are drugs that have been designed to treat patients in everyday situations rather than in cancer. There are some examples which are very common and which are receiving lots of investigation in cancer at the moment. So aspirin is the perfect example. It's being used now in many clinical trials to treat cancer, not just prevent cancer, which is what people originally thought it might be good for. Metformin is a drug that is used every day to treat diabetes. That's also being now investigated as a possible cancer drug. The kinds of drugs that we're looking at in our project are less well known for their potential in cancer treatments. So one good example is a drug called cimetidine, which is a very commonly used antacid used to treat stomach ulcers and excess stomach acid. We have data from previous clinical trials where cimetidine was shown to improve survival for colorectal cancer patients when used around the time of surgery. We understand from laboratory data that cimetidine, although it's designed to tackle stomach acid, also has effects on the immune system, on cell adhesion, on many different pathways that are relevant to cancer. There is a huge potential, we feel, for using these drugs in the perioperative period. And there we do see a common pattern emerging in different cancers. The pattern consists of patients treated for their primary tumor with surgery, and then following surgery there is a very fast relapse, often with distant metastases. And we see this in data from breast cancer, non-small cell lung cancer, osteosarcoma. Many different cancers have this common pattern. And we believe that one of the reasons for this is that the surgical procedure itself initiates a wound healing response. After the incision, the body reacts. There's an elevation of stress. There's a release of angiogenic factors to encourage the wound to heal. There's a drop in immunity. So all of these things occur after surgery. And these same factors, this wound healing response, is actually very useful to a tumor. A tumor needs to sprout a new blood supply. So when the body releases these angiogenic factors to help knit the wound and sprout a blood supply to heal the wound, the tumor can use that as well. So we believe that using some of these old non-cancer drugs we can intervene at the time of surgery to interrupt that wound healing response and therefore deprive the tumour of these additional factors that it needs to survive and to sprout. So where there are micrometastases already in the lungs, you give surgery, all of these factors can suddenly contribute to that metastatic nodule suddenly coming to life, and that's what we want to stop using these old drugs. There are obviously scientific challenges involved, and we need to take the preclinical data that many groups have published and the data from old clinical trials, and we need to kind of update that with new randomized clinical trials to show again the efficacy of this approach. So that's the first challenge. We need to run good clinical trials with clear results that show that making these perioperative interventions has an effect on the recurrence rate. But there are also non-scientific challenges. Many of these drugs are off patent, they're generic. Clinical trials are very expensive, and without IP protection, a drug company cannot guarantee a return on investment, so they're not keen on running clinical trials. They can't make money from this. So it's a challenge to do this in a situation where we don't have that support. And secondly, once we do prove efficacy, drugs have to be licensed for a given use, and going for a new license requires an investment. It requires quite a large amount of time and money to do this, and again the pharma companies have no incentives to support us doing this. So there are regulatory and financial obstacles in the way of making this work. One of the ways forward, obviously on the scientific side, is to partner with clinicians who are keen to work with us in running the trial. So we're here at TMC talking to different groups of clinicians who have an interest in this. And so we hope that through that work and through the funding that we and other not-for-profit organizations can make the trials happen. At the same time, we're also involved in talking to regulators, talking to patient advocacy groups to try and build a solution to the non-scientific issues that we face as well. I think for the future we need to prove that this approach works, and we need to take an example drug and take it through from the clinical trial right through to relicensing. And there are some good candidates for that. One of them is a drug called propranolol, and there's been some fantastic work done in TMC here. We want to take that forward. We want to kind of take that as our template for how we can do this to other drugs as well, once we've got the data and once we've got the regulation sorted out.